HIV is a major global public health issue, having claimed more than 36 million lives so far. At the end of 2012, close to 28 million were eligible for antiretroviral therapy under the WHO 2013 guidelines. However, only about 10 million people had access to it. CD4 lymphocyte cells, also called T cells or T helper cells, are the primary targets of HIV. The CD4 count marks the degree of immunocompromise. It is used to stage the patient's disease, determine the risk of opportunistic illnesses, and guide decisions about starting and monitoring antiretroviral therapy. Access to CD4 testing, however, is a major challenge in providing treatment to millions of HIV-positive individuals in the developing world. There is hence an urgent need for a low-cost, point-of-care CD4 test which will enable access to treatment to millions of HIV-positive individuals worldwide. Mylan has partnered with Zeomix USA to enhance access to testing along with the treatment to those in need in limited resource settings. Presenting Myt4, rapid, affordable, accurate, a truly point-of-care CD4 test. The Myt4 CD4 test consists of a single-use, disposable test, electrical mixer spinner reader EMSR accessory with charger, and an EDTA-coated blood collection tube BCT. The Myt4 CD4 test is self-contained and stable at room temperature with all reagents contained inside the Zeosphere reagent pellet. The test consists of an inlet for the sample, a reaction chamber where the blood mixes with the Zeosphere, a passage separating two chambers, a sedimentation chamber where the CD4 plus cells spin into a stack, and finally a cell ruler that displays the quantitative result. Test procedure. The accessory is opened, the twist handle is unlocked by turning it clockwise until it releases up, and the accessory is turned. It is important to verify that the power button lights are green. The battery has at least one bar. There are no error indicator lights on. Once the EMSR accessory is ready, 100 microliter blood sample is collected in the blood collection tube BCT, either through capillary or venous blood sampling. A new Myt4 CD4 test is opened from the foil pouch. The blue cap sticker is then opened just halfway to expose the sample inlet. Step 1. Sample. The blood collection tube is inserted into the sample inlet of the test. While holding the blood collection tube inside the test sample inlet, the plunger is used to push the whole sample slowly out of the tube into the test. It is critical that the 100 microliter sample be accurately transferred from the tube into the test. If there is any leakage or the sample flows back into the tube, the test is discarded. The foil cap is replaced over the sample inlet. Step 2. Mixing. This green button is pressed to turn on the light A. The test is placed into the test holder at location A with the arrows aligned. The clear protective lid is closed. The mix process is started by pressing this green button. The mix process continues for 4 minutes. Within the Myt4 CD4 test, the blood enters from the sample inlet into the reaction chamber. Inside the reaction chamber, all reagents are contained in the Zeosphere reagent pellet. The Zeosphere contains heavy CD4 specific particles and magnetic CD4 specific particles. These particles are conjugated with anti-human monoclonal antibodies. Sample introduction releases reagent particles from the Zeosphere. During mix, the accessory shakes the test for a defined speed and time to mix and bind the reagent particles to blood cells. Then magnets remove CD14 monocytes from the sample. When the mixing process is complete, the light A turns off, light B turns on, and there is a sound. The clear protective lid is opened, Myt4 test removed from the test holder. It is important to check that the sample has not leaked below the cell ruler or out of the test, and to ensure that the Myt4 test is not turned upside down. It is strongly recommended to proceed immediately to the next step and to not leave the Myt4 test sitting in the test holder. Next is step 3, twisting stage. The black test release button is held down and the Myt4 CD4 test is placed into the test holder at B with the arrows aligned. The test is pushed completely down until the black test release button moves back up. The clear protective lid is closed and the twist handle pressed down all the way. The twist handle is then rotated clockwise to one complete 360-degree turn until it is released up. The twist handle turns the top of the Myt4 test, which opens a passage below the reaction chamber. This passage allows the CD4 plus T cells with heavy particles bound to them to move into the sedimentation chamber. Step 4, the spinning. The spinning process is begun by pressing the start button. The spinning process continues automatically for 4 minutes. The sedimentation chamber of the Myt4 CD4 test contains a high-density medium. The accessory centrifuges the test for a defined speed and time. Spinning isolates the CD4 plus T cells through sedimentation, causing the CD4 plus T cells and heavy particles to form a stack in the tube inside the bottom of the test. When the spin is complete, the light B turns off. The clear protective lid is opened. The black test release button pressed and the Myt4 CD4 test removed from the test holder. It is important to check that the sample has not leaked below the cell ruler or out of the test, and to ensure that the Myt4 test is not turned upside down. Step 5, reading. The Myt4 CD4 test is placed into the test holder in the reader. The Myt4 test is checked for internal quality controls C1, C2 and C3. For C1, there must be a control line from the bottom of the C1 box up to 10. The cell stack cannot be below 10. For C2, the cell stack must be a continuous line. There must not be any gaps in the cell stack from 10 up to the top of the cell stack. For C3, there must not be a clog in the funnel at the top of the tube above C3. If all 3 quality controls pass, the results are interpreted by reading the CD4 plus T cell count indicated at the top of the stack in the same way one reads a thermometer. Each ruler line is 25 CD4 cells per microliter. CD4 cell count more than 950 should be recorded as greater than 950. The results are recorded and the Myt4 CD4 test discarded. The Myt4 CD4 test is rapid. The test delivers accurate results in less than 10 minutes, which makes Myt4 CD4 faster than any other CD4 test currently in the market. Service-free. It requires no maintenance or calibration, no service contracts or reagent rental. Accurate. Delivers results equivalent to flow cytometry, the gold standard of CD4 testing. Affordable. Myt4 CD4 is very affordable too, with no cost to start or switch from existing platforms. Versatile. Myt4 CD4 test is compact and highly portable. It does not require consistent electricity or refrigeration. And above all, Myt4 CD4 is very easy to use. To overcome the challenges of current CD4 testing, change to Myt4. Myt4, an innovation, a revolution in HIV care, across the age, across the globe. Myt4, rapid, affordable, accurate, a truly point-of-care CD4 test. Developed and manufactured by Zeomix, marketed by Mylan. Contact Myt4 at mylan.in. www.mylan.com